Thank you so much for joining us. So thanks, everybody. We're going to get started now. Christian Jobin is obviously from the University of Florida Health Center in the Department of Medicine and the Department of Infectious Disease and Immunology. And he's here today to give us a very special lecture for this series on microbial-derived small molecules in the cancer progression and treatment. This was a topic that was requested by all of you. It was quite a broad request, but I think Dr. Jobin is going to do an excellent job of talking about this. And feel free, as always, to ask questions in the Q&A box, which you've already seen how to do that. And make sure that if you have any technical problems, you put that in the chat. So with that, I will turn it over to you, Dr. Jobin.
Thank you so much. Do you need my video on? It doesn't work, so it doesn't matter.
No, you don't need your video. You're fine.
Okay. All right. So I apologize for the late start. I mean, we got a National Cancer Institute designation, which is a big deal, and in the U.S. and all the politicians and all the leaders were asked to attend a press conference today from 9 to 10, and that was not on my schedule. So that's with this in mind, let's get started. I heard that you're interested in microbiome cancer, so here's a cartoon from a review written by the lab of Rob Knight in San Diego. And what it shows is basically a different group of bacteria associated with cancer. You have the cancer type here listed, and you have numbers here. Those are the numbers that correspond to reference within the review, and you have a color code. The color code refers to either the paper is a diagnostic paper, mechanistic, prognostic, or therapeutic papers. And you could see that some form of cancer have not much literature behind it. So that will be thyroid cancer in this case, for example. And other form of cancer has a lot of literature, which kind of makes sense. In the term of microbiome, a lot of research was done in the gut because that's the most predominant location for microbes in the body. Now, this span all the different aspects of diagnostic, mechanistic, prognostic, and treatment. And I will discuss in more detail colorectal cancer. Here's a roadmap that I built for this lecture. The first thing we're going to discuss is microbial genotoxins and development of colorectal cancer. I will follow this up with the role of microbiota responsiveness to treatment, and close the lecture with microbiota and immune checkpoint inhibitor. So 3 chapters. Let's start with the first one. This is a cartoon that we build illustrating the concept, and I think this is well accepted in the scientific community, that, for example, in the gut, these microbes that are present in our gut produce different molecules that impact the host. And this host has either an immune cells, different immune cells, mesenchymal, epithelial cells. At the same time, the host is producing molecules that influence the microbes. So you could see that this is a dialogue, a communication network, and it's a healthy communication. So it's a default communication for a lot of us who are healthy. This is functional. You need that interaction with your microbes, and it's established early on. As soon as you're born, you'll colonize vertically from your mom, and you become colonized by bacteria, and you develop a dialogue relationship that help you maintain health. However, this communication could be disrupted, and modifiers that are responsible for this disruption could be either inflammation. You don't have to be in a chronic, severe state of inflammation. It could be a subchronic inflammation. The way you are approaching nutrition, so diets, the medication that you take, and to some extent, the genetic background that you inherited. All these factors could change your microbiome, either at the planktonic level, which will mean in the luminal location, of the bowel, intestine, or the microbes that associate with your bowel. So the organizational aspect of microbes on your tissue. And this changes of microbial composition and interaction could create what we define here, a microbial carcinogenic network. So now you're not in this healthy state anymore. You are configuring your microbiome into a carcinogenic network. Which kind of network could that be? I'll give you some example here. Colibactin is a small molecule produced by some bacteria. I will come back to that. Cytolethal distancing toxin. It's another genotoxic bacteria molecule. B. fragilis toxin, another genotoxin called OSHA, or even Clostridium difficile toxin. So many of these toxins exist in your bowel. And they could foster neoplastic changes that lead to transformation and tumor development. So that's the carcinogenic network. And many papers have supported this cartoon and development of colorectal cancer, for example. Now, these toxins sometimes coexist. For example, patient with family adipo FAP colorectal cancer, genetic cancer, have presence of colibactin and B. fragilis toxin. And that has been shown in a science paper by Cindy Sears and Hopkins University, so that if you delete one of these toxins, you have less tumors than a single toxin. So they synergize together. So it's a soup, if you may say, of toxin that may promote cancer. I like to focus on this one, colibactin, because that's the one that we have been studying in my lab for quite some time. So it gives you an example how that could work. So this colibactin is mostly produced by a group of Escherichia coli. I think you all know Escherichia coli. It's a very large species. Some are pathogenic, some are pathobiome, and some are commensal. A group of B2 E. coli possess a genotoxic island called PKS that produced that small molecule called colibactin. It was discovered in 2006 by the group of Eric Oswald in France. And they were screening E. coli for phenotype. So they were infecting epithelial cells and looking at the size of the cells in the nucleus to see if there are genotoxin, and running cell cycle assay and cell division. And they could see that if you have a presence of the PKS island, your cell cycle is stopped at G2. And usually what happened at G2 is your DNA is damaged and the cell tried to fix it. So you stop proliferating and you start repairing the DNA. And if you don't have the genotoxin, then you're G1 and you divide and replicate and everything is fine. But if the toxin is there, you're stuck in G2. That was an important contribution. But no one knew exactly the role in cancer. We discovered 6 years later that some E. coli that are found in the human animal facility that we didn't know existed, but we found it in our colony, and we call it North Carolina 101 because I was at the University of North Carolina at that time. So what we discover is that if you associate a germ-free mouse susceptible to colitis, which is IL-10 knockout, which is an immunosuppressive molecule, if these mice that are healthy and germ-free, but then if you introduce this NC101 PKS positive, which are capable of producing colibactin, or isogenic mutant NC101 that is deleted for PKS, so there's no genotoxin capacity, then you look at the tumor development, this is number of tumors, you could see that the mice that associate with E. coli that cannot produce colibactin have less tumors than the one that could. And if you have tumors then these tumors are less invasive. So you have less capacity to promote metastatic tumor, for example. So that was a proof that this PKS island is important for the development of colorectal cancer. It took a long time to identify and solve the structures. Again, this is an island. I will come back to that, a genotoxic island that produces a small molecule called colibactin. It took a long time to solve that structures. This is a predicted structures of colibactin. And what you could see here is a structure called a cyclopropane here on each side of the molecule. Cyclopropane are highly reactive to DNA and will cause interstrain cross-linking. So each strain of the DNA is clamped or trapped by these two cyclopropane and basically create a stall, fractures in the DNA that has to be fixed, okay? What do we know about the function of PKS in terms of cancer? Well, a little. This group in Holland from the research group of I'm blanking on the name here now. It's kind of embarrassing. But what they did, they injected the PKS positive E. coli inside an organoid, human organoid, okay? And this E. coli will produce eventually colibactin. And after multiple rounds of exposure, they isolate the DNA from the epithelial cells and did whole genome sequencing to find mutation. So if the colibactin is important to create mutation, you should be able to sequence and see these mutations. And what they found is that there were this PKS positive E. coli was able to produce a very unique signature called SBS, single base substitution 88. So it has a unique signature, and also a short indel called indel or ID18. So those are unique because they were never found before, and they were able to see that in the organoid, human organoid in vitro. And they searched for that signature in colorectal cancer database from the TCGA, the Human Atlas, Cancer Atlas, and they were able to see that in a small proportion of colorectal cancer patients, 5 to 8% of human having colorectal cancer shows these signature, implying that it could be important or may have been the cause of cancer. We don't know for sure. Okay, so now I told you about the PKS island. This is the island. It has 19 genes, all right? And a lot of them are encoding for enzyme. And these enzymes are building a compound. It's the same way as penicillin is produced, or rapamycin. It's called a assembly line. So genes will produce an enzyme. The enzyme will start building a compound, transfer it to another enzyme, and so on and so on. So it's a factory lane assembly. And you end up with this molecule. And this is an inactive molecule because it has this long lipid tail on each side. So that molecule is not active. So it's a prodrug, not activated yet. The island has its own activating enzyme called CLBP, which will cleave this long lipid tail here and here, and that will give rise to the active colibactin with the cyclopropane motif I told you about. This is armed and fully functional. We don't know the function for the bacteria, but we know that if this come in contact with DNA, it will break and damage DNA. Let's take a look inside the bacteria now. Okay, so I told you this is mostly produced by E. coli, and the island looks like that. So let's take a peek inside an E. coli and look how it works. All right, so this is my PKS genotoxic gene island, okay, with the 19 genes. This is in the cytosol of the bacteria. The bacteria, like E. coli, has an inner membrane and an outer membrane and a luminal space called periplasm. And this is the outside, okay? So that will be outside of the bacteria. I told you that the PKS island will produce a prodrug. It's called pre-colibactin. It's not activated. It could be activated by an enzyme called CLBP, but the enzyme, the active motive of the enzyme is facing the periplasm, right? But the molecule is in the cytosol. So how do you get this going? Well, we discover that the island is encoding for a transport molecule called CLBM. It's a mate transporter. It moved the pre-colibactin inside the periplasm. Now that the molecule prodrug is close to the enzymatic activity of CLBP, it could be cleaved and fully activated. And the reason for that is you don't want the molecule to be activated inside where the DNA of the bacteria is, because the bacteria will likely die. They don't have a repaired system. So it's a clever way to move the drugs away from the DNA and activating the drug in a safe location. Now, how is this drug going out? No one knows. We don't know if it's transported. The only thing we know is the bacteria has to be in close contact with the target cells. This compound is not, has never been purified in the supernatant of bacterial culture, could not be found. It's extremely unstable because of this diketones motif here, so it breaks into extremely unstable. So what happened is likely that the bacteria attach, because this type of E. coli attach to mammalian cells, and that compound could be transported. We don't know how, inside the mammalian cells. What we know is the cyclopropane motif bind to adenine of the DNA on each strand of the DNA and cause interstrain cross-linking. Okay, so let's revise this for a moment. I told you that the proof that the PKS is producing or is damaging DNA and producing mutational signature came from this PKS E. coli infection in organoid, which is intestinal epithelial cells culture in vitro. And that's how they found this mutational signature, SBS 88 and NDEL 18. But the real question here is, what's the mutational burden of PKS positive E. coli tumors in a real tumors? This is not a tumors, this is a cell system. So we were interested in understanding that. And to address this question, we did a very simple experiment. We used germ-free mice. So we have access to germ-free mice. I'm sorry about that. I'm going the wrong way. I'm using a phone system. Okay. All right. Using this system, it's a germ-free mouse. APC-min mouse are genetically susceptible to small bowel polyps. APC mutation is found in 80% of sporadic cancer. So it's a very relevant model, but it's mostly developing cancer in the small bowel. But when we expose these mice to dextran sodium sulfate, which is inducing some wounding of the large bowel, then the tumors, you see a significant increase of tumors in the colon. And because they are germ-free, it's easy to introduce our bacteria NC101. So we introduce NC101 that has PKS positive or the strain that doesn't have the enzyme that activate the drug. So one is able to produce calibactin, the other strain is not, okay? And what we did, we let the system go, and you could see here by pictures, number of tumors here. And what we did, we collected these tumors, okay? And also collected the tissue here from the non-producing calibactin mouse. And we confirmed that they are tumors. You could see the number of tumors increasing if you have NC101. If you don't have active calibactin, you have less tumors. And we sequenced that, okay, whole genome. No, this time we did exome sequencing, just the messenger RNA.
So after flag, push with the line. After flag.
Is there a question, or there were a lot of noise? I'm not sure if there's a question. And I don't see a chat, so I'll keep going. So what we did here is that we were able to exome sequence tumors and off tumors. And then by calling a single nucleotide variant, we could know if there is a mutational profile. And what we see here is that you could look at increase SNP number if you have an active E. coli that produce calibactin and less. And we just study mutational signature, and what you could see, there's a different pattern here. The only one signature that survived statistical analysis is SBS15, which is a signature associated with mismatch repair defective. And you could see here, if you have the NC101 infected mouse and you look at the tumors, they have an increased signature of SBS15 compared to the non-calibactin producing. So the SBS15 has is characteristic of tumors produced by NC101. So that was an interesting observation. And you could see here that the number, this SBS15 mutation is predominantly seen in colorectal cancer. This is the prevalence. It's the highest prevalence compared to other form of cancer. Now we have a different, well, there's two aspects here. This signature was obtained by infection with the bacteria producing the PKS and was done in the epithelial cells in vitro. I've shown you that there is a mutational signature if you do the bacteria in tumors, but this signature here was done with the bacteria, not the molecule, not the calibactin. No one has purified calibactin. So there is a structure-function relationship missing to understand how this calibactin could induce cancer. So we partner with the group that solved the molecule structures in silico. All right, so this is not purified, this is deduct chemistry. The PI is Dr. Seth Herzog at Yale University. We collaborated because they synthesize this molecule. They are chemists, so they were able to synthesize that molecule that is really similar to calibactin with the, or identical to the predicted structures with the cyclopropane motif here. And we had a negative molecule where the cyclopropane is unlinked by methyl groups. So this one is predicted to be inactive because the only difference here is the cyclopropane is missing compared to the active one. And with that, we could study structure-function relationship. We did different things, but I will skip a lot. And that was driven by my student, Michael Daugherty. And what it did, he used different amount of this molecule and expose human organoid to the molecule, active or inactive, and then stain for a histone 2-AX, which is a marker of DNA damage. Remember, I told you this molecule with the cyclopropane bind to adenine of the DNA and cross-link DNA. And the cells need to resolve this cross-link. And what it does, it cut the DNA, creating a double-strand damage. And this gamma H2AX is a marker of DNA damage. This is mitomycin C. It's a positive control. You could see the organoid staining for gamma H2AX. This is an overlay. And now this is the active molecule, different concentration, 10 to 100 micromolar. And this is the inactive compound that fails to induce DNA damage. And this is quantified here. So the molecule itself could induce DNA damage, and it's dependent on the presence of the cyclopropane. Now, we wanted to know which kind of gene expression we have and which kind of DNA damage we will see, because now we have the molecule. So we did culture cloning of HCT116 is a cell line, colon cell line. We expose this cell clone to active colibactin or inactive. We repeated this exposure 10 times with a 40 hours treatment and 40 hours recovery. Then subclone that to have single cell clone, and did RNA sequence for gene expression or whole genome sequencing for mutational pattern. This is the DNA, this is the transcriptional profile. Every single dot is a different clone that we produce that are exposed to the active molecule or inactive. You could see the transcriptome by PCA, quite different. And this transcriptome was measured 30 days after exposure. So this is a memory imprinted in the cells that makes it very unique in terms of transcriptional profile. And if you do volcano plot, you see some genes. This is P50 cytochrome that is induced in detoxification of chemical compound exposures, some inflammatory genes, and some DNA repair genes. These downregulated are associated with P53, which are pro-apoptotic. So you downregulate apoptosis here and upregulate DNA damage and inflammation, basically. And this is a IPA looking at pathways analysis. And you could see again that many of the pathway induced are involved in DNA repair, which makes sense. I told you there is a DNA damage induced by this cyclopropane containing molecule, and the cells try to resolve it. So far, the data indicate that this molecule induced DNA damage, changed the transcriptome relevant to DNA repaired and pathways. Now, since we have the DNA from the same cells, we look at a mutational profile. And again, this is a number of single nucleotide variant. So the active compound induces a lot of mutation point compared to the inactive. And if you break down the contribution, you could see that there is a different type of signature. It's a composite signature. SBS 88 is the one that was observed with the PKS E. coli from the Hans Cleaver groups in Holland. So they were using organoid and the bacteria. Now we're using a molecule, and we show the same one to 25%. This one is a mismatch repair deficiency. You see this signature SBS 44 when you have mismatch repair deficiencies, which is often found in colorectal cancer. 17 is due to radical oxygen species exposure. These two has no etiology, so we don't know where they're coming from, what's the reason for them. But these three we know. So mismatch repair, I remind you, was found also in the tumors of the mouse exposed to E. coli. it was SBS 15. Now here with the molecule, we also have mismatch repaired. And we look at NDEL, and we found the same NDEL signature as Hans Cleaver's NDEL 18 they found in the organoid. We found it with the molecule. So the molecule somehow overlap with what the bacteria is doing, at least in vitro, some SBS 88 and NDEL 18. Now, because we found the SBS 88 and, like I mentioned here, you have 44 and we have 88. So the mismatch repair coexists with SBS 88. We wanted to know the relationship with this coexistence way. So we looked at the database to look if they were associated with each other. And this is the data that are significant. We found SBS 88 associated with SBS 1. And this is a mutation that is increasing with age. So the more age you are, the more of this SBS 1 you will find. So this is a clock system. So it's interesting, but not highly relevant to us. 18 is a radical oxygen species mutational signature that associated with SBS 88. And here we have our SBS 88 and 44 that associate together. So this is from a database of close to 300 patients when we looked at these coexistence. Then we extracted just the database that is relevant to cancer. So there was close to 300 or so. And we could see that there is a increase SBS 44 association with SBS 88. So these two signatures coexist. What does it mean to have mismatch repair deficiency if you bring an E. coli PKS in the system? So this experiment addressed that, because so far what I've showed you is that there is coexistence of SBS or Colibactin with mismatch repair. But does it cause more cancer? To address that, we use the EPCM in mouse that I described before, but it will cross with mismatch repair deficiency 2, MSH2. The flux means it's tissue specific. If you cross it with a Cre recombinase that is expressed in epithelial cells, then you delete that gene only in intestinal epithelial cells. And then we introduce the NC101 functional or the delta CLBP, the one that is not producing Colibactin. And you could see here the increased number of tumors when MSH1, 2 is not present compared to when the mismatch repaired is present. So you need the mismatch repaired system to fix what Colibactin is doing to the cell, right? That's the bottom line of this experiment. And you could see here the number of tumors increase if you are deficient in mismatch repair. So this data indicate that Colibactin cause DNA damage that has somehow been resolved by mismatch repaired system. If you don't have that mismatch repaired, you have a higher likelihood to develop colorectal tumors. So I'll close this section here just by saying that the PKS positive E. coli has clinical application. Maybe it could be used for preventive screening. 60% of colorectal cancer patients have PKS positive, but 30% of healthy people do have PKS positive E. coli in their bowel. Should we screen these people for risk? That's a question. Here, a lot of anti-tumor drugs are inducing DNA damage, right? So they could be used to kill tumors. So the question is, could one build a Colibactin that is killing tumors? And there's people working on that. And the other thing is clinical intervention. Should we delete this E. coli PKS by using precision microbiome editing, for example? And that I mean bacteriophage that are specific to these type of E. coli to eliminate a risk factor, or a synthesized inhibitor of colibactin, which already been done so far and works in preclinical models. So this is the clinical side. In the basic science, we are not sure what's the DNA damage response pathway induced by colibactin. We don't know how this molecule get into mammalian cells to cause DNA damage and potential cancer. And we don't know why the bacteria is producing this colibactin. We have no certainties of the function of this compound. So a lot of research is still needed. All right, so I'll move to this. These are related. Microbiota responsiveness to treatment. This is the work of Dr. Ye Yang. And I've shown you this cartoon. I told you that this is healthy relationship. Modifier could come to disrupt this healthy relationship. I highlighted inflammation here, okay? And that caused the creation of this carcinogenic network, and all these toxins may be activated. This is very important. So the question is, if, let's say, inflammation is important to create that changes here, could you intervene and block inflammation to prevent the creation of this carcinogenic network? We addressed these questions, and what we did, we used an anti-TNF antibody. You know, the same as what people that have rheumatoid arthritis or psoriasis, they are on anti-TNF medication, okay? So let's block inflammation and see what happens. Now, I told you about the APC-min mouse, so you're familiar. What we did, we didn't use a germ-free here. We exposed the mice with antibiotics, so it would be easier to put our NC101 that produce colibactin. We did the DSS, as I mentioned before, and we treated the mice with anti-TNF or PBS, 6 injection, and looked for cancer development. And what we found is the anti-TNF decreased tumors, and you could see the tumor here, pictures, and that's great. Right? So the TNF works in preventing tumors. We don't know the mechanism, but what we thought, because I told you inflammation changed the microbiome, we think, right? So we went to look at the microbiome over time, because we could follow these mice and collecting their stools at day 0, 14, 35, and at the time of euthanasia. And this is a microbiome study where at day 0, the mice receive no treatment. They are coded red and blue, but they did not receive any treatment. And the microbiome is not different because they are the same genetic background. So you don't expect microbiome to be different, and they are not. At day 14, this is when mice receive 3 doses. They're already at the 3rd dose of anti-TNF. And you could see the red mice, every dot is a single microbiome of a mouse, and the blue is non-treated. So you could see that the microbiome of the cohort TNF treated is quite different than the one that is not treated. And you look at day 35, which is after the 6th dosage of TNF, anti-TNF. Look at the microbiome, very different. So the mice receiving TNF have a different microbiome, not only in the stools, but we collected colon tissue. And in the tissue, it's also different. Right, so that's great. We look at the transcriptome of the microbiome. I told you that a carcinogenic network is probably formed by inflammation exposure. This is day 14. This is a stool of 4 mice on each cohort. After TNF treatment, you see the transcriptome of the bacteria quite different. This is metatranscriptome. and at day 35 as well. All these data were normalized for whole genome, metagenome. And a reason why you may have less tumors when you treat with TNF, anti-TNF, is because the E. coli PKS is maybe decreasing, right? So you put NC101, you treat with TNF, anti-TNF, and then E. coli disappeared. But that's not the case. This is the whole genome, metagenome, looking at NC101, and you could see there's no difference between treated and untreated throughout the day. So the E. coli is there in the bowel of treated and untreated. It is just not causing cancer. Which genes are different? This is a heat map of genes. Most of the genes that we looked at were downregulated compared to untreated, except two here that are phenylalanine metabolism and aromatic compound. But most of the genes were downregulated, as well as day 35. The pink means that we looked at data set of IBD patient treated with anti-TNF, because those are published, and they overlap with our mouse treated. So there's some significance to that. Now, I told you that microbiome changed when you treat it with anti-TNF, but is that meaningful? Is the change leading to any phenotype? So we did a simple experiment in the microbiome field. It's called co-housing. So we put a mouse that is not treated or a mouse that is treated in the same cage. Since mice are coprophagic, so they eat each other's feces, the microbiome is transferred from one mouse to another. Now, remember, the mouse is treated, the same mouse is treated with TNF, okay? So they receive the treatment. But look what happened. The anti-TNF did not block tumors. So you have the same amount of numbers of tumors. The only thing that changed in this experiment here is the mouse share the same cage, so they are eating each other poop, which suggests that the microbiome was very important for the anti-TNF effect. And look at the microbiome, the red and the blue, no difference. So before I told you they were very different with the different cage. Now it's the same cage, and the anti-TNF is, the microbiome is not different, even on the tissue. And that failed to move or to block cancer. But that's an interesting observation. But the real one will be to take the microbes of a mouse that is treated with anti-TNF and put it in a new mouse that has never received treatment. And that's what we did here. We took fecal content of a PBS treated or anti-TNF treated, put it in a new cohort. These mice never saw the treatment. These mice are just receiving microbiome from different cohort. And we did the same. So it's an episemin mouse exposed to DSS. And look what happened. The anti-TNF treated microbiome put in a new mouse. That mouse developed less tumors than if the mouse was populated with a regular microbiome. So the memory of the treatment is transferred to a new cohort. This suggests that when you have a microbiome that is exposed to inflammation, it changed. I told you that, right? So the microbiome changed and create change in the functionality of toxin that cause neoplastic changes. If you intervene to block this inflammation, you're likely changing this carcinogenic network, and now you have less tumors. So the microbiome is very important in the therapeutic impact. So that brings me to this. Carcinogenic bacteria per se is dependent on the environment where it evolved. If this environment has metabolic changes, so increased spermidine, increased glucose different level of iron. It all impact, for example, PKS gene expression, which could impact Codibactin production. At the same time, looking at inflammation, we told you anti-TNF is probably, we don't know the mechanism here, how it decrease PKS, but mesalamine, which is an non-steroid anti-inflammatory drugs, block polyphosphate kinase, which is very important to induction of CLB, which is the genes in the PKS island and increase gene expression. So there is a relationship with the environment, either inflammation or metabolic, that impact the activity of bacteria and consequently neoplastic changes. That brings me to the final chapter, which is bacteria immune checkpoint inhibitor. Now, so far, I've been telling you about microorganisms that promote cancer and how it may work. Now I will tell you about microbes that help drug responsiveness. This is an interesting group of papers. I covered papers, 3 manuscripts that were published in Science. What these people have shown is that the immune checkpoint inhibitor PD-1, there's different responsiveness in patient. There is maybe 20% responsiveness depending on the form of cancer. We are not sure why patient respond or don't respond, or the drug fail or not fail in patient, but this group of people decided to look at the microbiome of patients that don't respond to PD-1 versus responder, and they found there were different microbiome. So responder has a different microbiome than the non-responder. To look if this is functional, they transfer this biome into a mouse that has the same type of tumors implanted, and then treated with the same drug as what the patient, the PD-1 blockade. And what they observe is that if a mouse has a microbiome from a human responder, they kill the tumors better, and they associate with an increased anti-tumor CD8 positive T cells. And the mouse that has feces from a non-responder patient have an increased T regulatory cells, which decrease inflammation and have poor anti-tumor response. All these papers were published in the same issue of Science. Although they all observe the same thing, they have different bacteria that they think are responsible. For the Paris group, Akkermansia. For Texas, Faecalibacterium, and from Chicago, Bifidobacterium. These bacteria are very different from each other. So why don't they have the same signal? Because they have the same phenotype. We were very interested into that, and we downloaded all the data that this group published, and we ran machine learning, okay, so a form of artificial intelligence. And the data were assembled because it's whole genome, metagenome. We could assemble them to define microbial species. And we run 3 different machine learning: random forest, support vector machine, or lasso. And a predictor of response, if you were looking at a biomarker, the area under the curve should be top left corner for high sensitivity and specificity. You could see here that there's nothing is formed. So there's no predictor whatsoever across the data set. None of these data could tell you that the specific macros will define responsiveness. But if we use the same data set but assemble, because they are genes, right? We could assemble in functional pathways. If we assemble the data in functional pathways, you could see a curve here somehow been built for the Texas group and the Chicago group, but not for the French group. But again, this curve is not clinically relevant. It's too low to build any clinical data out of that. But it's indicate that functional activities may drive responsiveness, not the specific type of microorganism, but what they do. Even if you have 2 different microorganisms, they may be inducing a similar response on the host. And this is all the data published so far. in microbiome immune checkpoint interaction. Okay. Now, this is the French group I told you. This is non-small cell lung cancer treated with PD-1, and patients that respond have an increased Akkermansia municiphala. So that's the bacteria they identify. But look here, this is a group from South Korea, published 2 years ago. What they found is that Akkermansia municiphala is associated with non-responder, the opposite of the French group. Again, there is a lack of understanding and clarity. So for that, we were interested in looking at the microbiome of non-small cell lung cancer in a US cohort. Those are Korean cohort and a French cohort. So we partner with Moffitt Cancer Center here in Florida, enrolled 65 patients, collected feces at baseline. This patient went into a immune checkpoint inhibitor treatment, mostly PD-1. And then after a treatment, responsiveness was assessed. So we got, again, 20% of responder. And we collected some of these specimen, fecal specimen in preservatives, so the bacteria are alive, okay? And we published that last year, so I will be brief on that, but what we found is that, similar to the other group, if we take non-responder feces, put it in the mouse, or another cohort, a responder feces, put it in the mouse, these mice are germ-free, so they will stay humanized in terms of microbiome, so they will not be contaminated by other murine bacteria coming from a facility. We waited 2 weeks for the microbiome to implant, and implanted the Lewis lung carcinoma, so it's a very difficult cell, non-responder to PD-1, and then treated the mice with anti-PD-1. And this is what we found. If a mouse is colonized with non-responder feces, it has more tumors than if the mouse is colonized with responder human feces. So we reproduce basically what the other group have shown, okay? And the idea was that, just to make sure it works, and then study the microbiome. This is an analysis at the ASV level, which is amplicon sequence variant level. Every dot is represent a species from this genus here. Above the red line means it's enriched in the responder. Lower than the red line means it's enriched in the non-responder. What you could see is that here's the Akkermansia group that the French and the Korean group identify. We have one species that associate with responder and one species that associate with non-responder, exactly what these two groups publish. So what it means, it's very complicated, okay? And the most abundant bacteria that we found in the mouse that associate with responder or non-responder is Bacteroides. So it became clear to us that genomic would not solve the problem. What we needed to do is to isolate bacteria. And for that, we utilize this system. This box here is called a prospector. We house this in an anaerobic chamber because most of your bug, 95% are anaerobic. So you don't have any, they don't tolerate oxygen. So this is a best system to keep them happy. We took feces from responder patient and use that system. What that system does is that there is an array that is composed of 6,000 nanoliters wells. And we dilute the feces in a way that we could load this array, and all these individual wells could be populated by one bacteria, okay? And the idea for that is that if you have only one bacteria per well, they will not compete with each other for food, and you have better chance to isolate bacteria. So that's what we did. These wells contain a molecule called rhizofurin, which is fluorescent depending if the molecule is utilized. Okay, so it's a metabolic tracer. If the bacteria are there, fluorescence will change. The computer of the machine will pick the signal and will array that. Once we have array, we transfer that into a plate that we could use for identification using MALDI-TOF biotyper. Okay, so this is a sophisticated mass spec. And we have a database that could identify the species. And if it's not there in the database, then we sequence the entire 16S genes of the bacteria and sequence by Sanger, so we could have an identification. I told you that a lot of Bacteroides were found in our responder bacteria, so we were interested in these guys. So we found a lot of bacteria. Now, since we are interested in anti-tumor response, which means that immune cells likely that kill tumors will secrete anti-tumor molecules. So what we decided to do is to take the 183 Bacteroides that we identified by the prospector, plate them individually, one by one, into a 96 well plate to grow them. Then we took the supernatant of these bacteria and put it in contact with splenocyte isolated, CD8 positive T cell isolated from murine splenocyte. Why CD8? Because I told you those are anti-tumor cells and they mostly produce interferon gamma. So interferon gamma is our screen. So we screen all 183 bacteria in triplicates to find which one induce interferon gamma by our CD8 T cells. That was long, but was fruitful, and we observed only 3% of all the isolate we got could stimulate interferon gamma from the cells, and they were here. So we were happy with that. So what we did, we took the 6 isolate that produce or induce interferon gamma and form or build a consortium of bacteria. And we have 6 of these microorganisms here that match genetically these guys. So we put them together as inactive because they were not able to induce interferon gamma. So active consortium, inactive and active. And we screen these guys again, instead of a filtration at 22 microns, which basically remove bacteria, we subsequently filtered for small molecule, 3000 cutoff. So we were interested in small molecule produced by bacteria, and we screened for interferon gamma again. And what we found using our splenocyte CD8 positive cells, this is a positive control PMA inomycin interferon gamma production. This is our consortium responder before the filtration at 3K, and this is after filtration at 3K. So we still have bioactivity in the small molecule compartment of this filtrate. And this is the non-responder. So the non-responder consortium do not induce interferon gamma. So we were very happy with that because now we know it's a small molecule. And we took the consortium and did the same experiment I told you, took a mouse, put the responder feces or the responder consortium that we build or the non-responder feces and associate that with mice and then treat with anti-PD-1. You could see here the tumors with the non-responder feces human, tumor growth. And now the responder feces, tumor shrink. I've shown you that before. But now here's our consortium, only 6 bacteria. And you see, we have a decreased tumors as much, almost similar to the complex feces from humans. So now we have only 6 bacteria that reproduce what complex thousands of bacteria are doing. and more. There's viruses there. It's very complicated. So this is great. So we know that we have a magic potion, right? And since we have supernatant, we took an isolate from the 6 consortium, this guy, because it was inducing high amount of interferon gamma in vitro. We took this and started doing a bioassay guiding fractionation to identify the molecule, right? So we first did the vacuum liquid chromatography, 6 fraction, tested these fraction in our screen, and we identified fraction 6 as the only fraction that could induce interferon gamma. We took that fraction, put another round of fractionation through HPLC into 12 different fractions, screened that again in our cell system. And what we found is that this is our PMA control, positive. The only fraction that we found to induce interferon gamma was fraction 11. So we were quite pleased by that. And we took a large scale approach, culture leaders of this bacteria that produce that small molecule, and did NMR mass spec, which is more precise for molecule identification. And we were able to identify these molecules, okay? That one here, 430 mass spec, mass ratio, was very interesting because our 3 different identification pipeline agreed that that was the molecule structure. So we synthesized that molecule, right? Synthesize this molecule that exists in 2 different conformations, cis and trans, and use it in our screen again. So this time we don't use supernatant anymore. We use a molecule we identify within that supernatant. And again, this is our unstimulated. This is our positive control for interferon gamma production. This is our cis structure molecule. And you could see that different concentration increase interferon gamma production. This is the trans. Different concentration fail to induce interferon gamma. And when we combine cis and trans, we have a synergy that is higher than our positive control. So this molecule is able to induce interferon gamma. So in summary, to finish that, I told you that microbiota from anti-PD-1 responded patient, if we put it in the mouse, we have an increase in interferon gamma CD8 positive T cell. We could identify bacteria that when we put in a mouse, we have decreased tumors. We know that this bacteria from that consortium produce a small molecule that induce interferon gamma, well, the supernatant. And we were able to identify a small molecule that is responsible for this effect. Now we have a lot of unanswered questions. How is this molecule itself capable of inducing anti-PD-1 response? This is, is it anti-tumor on its own, or do you need anti-PD-1? Where is this molecule release? In the gut? Is in the gut, or the bacteria goes somewhere else to produce the molecule? We don't know. Which cells is the target of the bacteria and the molecule? And what's the molecular mechanism by which this molecule induces interferon gamma? So we don't know the answer to that. So we're working on that. But what I've shown you today overall is that you have a relationship, immune cells, tumors, that people know in cancer is very important. We have drugs and tumors interaction, right? If you think about responsiveness. And I told you microbiota and tumors, either development or blockade. But you cannot see this as an silo, because all these are interconnected. Microbiota and immune cells are interconnected. Microbiota and drugs are interconnected. So this interaction is very important to define and build clinical trial to test responsiveness or interaction between microbiome, drug, and tumor responsiveness. And this is the last slide, but the idea is that by identification of bacteria that may be associated with cancer development, one could, using the purple arrow here, manipulate the diet to change this microbiome, pre and probiotics. Bacterial therapy will be fecal microbiome transplant, design drugs that target microbial genes, or even design bacteriophage that specifically take bacteria that may be deleterious to a patient. On the other side, with the red arrow, you could harvest information from the microbiome to predict drug interaction or generate a biomarker of disease progression, responsiveness, or susceptibility to cancer. So this is very active in terms of research. With that, I will stop and just highlight the people who work on, I think I highlighted everyone that was important, directly involved, Yale University for the small molecule work, Raphael for the signature, Rad Garaby for all the microbiome analysis here at UF, and obviously all the fundings that were supporting this research. And with that, I think I'll thank you for your attention, and maybe we could have some, maybe you have some questions, and I'll be happy to answer that. If any. Hopefully, I still have you. Mr. Milner, are you still there? I have a queue here, okay. Well, that's not a question for me, but someone is asking if you get a video recording of all the session. I'm not controlling that. Okay.
Hi, Christian. Sorry, I was trying to get off the call I was on. Thank you so much. I think we are finished with the session, and it doesn't look like we have any questions. There is one from Dr. Sadiq. Did you answer that one? Can you shed more light on funding?
I'm not sure what it means. Fundings typical fundings include writing grants to federal or, local agency, private foundations, or support from an institution, depending on the, research interests. So funding's required writing grants most of the time for people like me, and being scored and ranked by my scientific peers, and then the funding decision belongs to the funding agency, whether they are federal or in the US federal or a national, and states, and then you hope for the best. That's basically.
Absolutely. Well, thank you so much. And there was another question about with the recordings. The recordings are all on the landing page. So where you registered to attend this session, that landing page has all of the recordings after the sessions are over. If you need the link to that, I will share that with Gloria so she can send it out to all of the participants when we conclude. And if you remember, please, from earlier in the session, I told you that we do not have a session on Thursday. We do not have a session next Tuesday, but our last session is June 29th. That's going to be our makeup session for our neuropathology section, which is going to be very exciting. That will be the last one. So please, if you have not already, please register for June 29th, the same time as always. And that will be our last one, and we're really excited about it. So thank you so much again, Dr. Jobin and Gloria, for joining us, and we will see all of you next Thursday. Not Thursday, but next Thursday. Okay, bye-bye.
Bye, thank you.
Bye, Dan.